Welcome to our MHC Tetramer webinar. Thank you for taking the time to join us today. We hope you will find this information useful. MHC Tetramers are a gold standard technology for monitoring immune cells involved in various human diseases and have also proven useful in mouse disease models. Before we begin, let me emphasize the take-home message from this talk. MHC Tetramers are the gold standard tool for monitoring immune cells in various human and mouse diseases with high specificity. So, what is the advantage of our technology? Our patented MHC Tetramer technology reduces nonspecific binding, resulting in clearer data compared to our competitors. Product availability. We supply over 800 items related to MHC Tetramers. If you cannot find the tetramers of your interest, we also provide a made-to-order service where we can customize MHC Tetramers to fit your needs. In addition, we can help you build your own specific tetramers using our new DIY kit called Quick Switch. MBL tetramers have long been considered the industry gold standard. Therefore, we are confident that we can provide comprehensive support for your research using MHC tetramers. The aim of this presentation is to help you achieve a deeper understanding of the MHC Tetramer technology. This is the agenda, and the whole presentation will take 15 minutes to complete. Questions are welcome and can be submitted to our website. First, we will briefly discuss the role and composition of the immune system. Our body is constantly exposed to various pathogens and other harmful materials. In some cases, people develop tumors. It is the primary role of the immune system to counter these pathogens or cancers and keep our body healthy. The immune system comprises different immune cells. These include neutrophils, macrophages, and dendritic cells, which are phagocytic. They engulf and digest bacteria invading the host cells. In addition, T or NK cells kill virus-infected and cancer cells. Antibodies secreted from B cells bind to pathogens and tumor cells to remove or detoxify them. Among these immune cells, T cells attack cancer or infected cells directly. As shown in this slide, T cells recognize these infected cells as dangerous, and this recognition activates them to attack and kill their targets immediately by secreting cytotoxic factors. This slide shows how T cells recognize target cells. Each T cell has a receptor on its cell surface called the T cell receptor, TCR. Every cell, including cancer cells, expresses an antigen and MHC molecule complex on its surface. Each peptide MHC class I complex is composed of an alpha chain, non-MHC beta-2M, and antigen. In the case of cancer cells, the antigen peptide loaded on the MHC is a fragment of multiple proteins expressed inside the cell. TCR recognizes each individual complex and assigns cellular identity. Monitoring antigen-specific T cells is very important and helps clinicians to evaluate the status of various diseases or patient response to treatment. The question that next arises is the following: how can we detect antigen-specific T cells? To answer this question, we will now discuss MHC Tetramer technology in more detail. Since MBL has more than 40 years of experience in developing antibodies we decided to use this expertise to develop novel tools to evaluate TCRs. Initially, we focused on developing specific antibodies to target each TCR. However, the nature of TCRs made constructing a specific antibody difficult, and we encountered challenges, such as the preparation of proper immunogens and a high degree of similarity among different TCRs. Consequently, antibodies against individual TCRs had very high cross-reactivity with other TCRs. Thus, this approach was too complicated and time-consuming. Given these considerations, we changed the strategy we were using to capture target T cells and acquired MHC Tetramer technology. In the following slides, I will explain MHC Tetramer and its application in this type of study. Over 20 years ago, researchers generated a recombinant peptide MHC complex conjugated to a fluorochrome in vitro. They then applied this monomer of the peptide MHC complex for detecting T cell targets. Nonetheless, the binding activity of the monomer TCR complex was too weak to facilitate detection of target T cells. The monomer was easily detached from the T cells. To overcome this difficulty, the monomer was biotinylated and tetramerized using fluorochrome-labeled streptavidin. It enabled the tetramer to maintain its interaction with the TCR on antigen-specific T cells. Here, we show the binding kinetics between TCR and the peptide MHC complex. The monomer of the peptide MHC complex sharply binds to the TCR but immediately detaches, while the tetramer binds to TCR very tightly and rigidly. Therefore, tetramerization is a valuable tool for detecting the peptide MHC complex, making it useful in T cell monitoring. This is how we synthesize MHC tetramers. First, we mix MHC peptide and beta-2M to make a monomer of the MHC peptide complex. Next, the MHC is biotinylated and purified by HPLC. Finally, the biotinylated monomer is tetramerized with fluorochrome-labeled streptavidin to form the MHC tetramer. So, how can you use the MHC tetramer? If you wish to analyze circulating T cells, it is extremely rapid and easy to use. First, collect whole blood, add the MHC tetramer, followed by a CD4 or CD8 antibody. Next, lyse the red blood cells and fix the cells. Wash once with PBS. Now you can analyze the sample using flow cytometry or fluorescent microscopy. For performing flow cytometry, we used two-color analysis. The double-positive cell population represents the target antigen-specific T cells. We will show you an example using a human sample in the next few slides. Here is an example of tetramer application in infectious diseases. This sample was obtained from a patient infected with influenza. You can detect the influenza-specific T cell using an MHC tetramer for specific flu antigens. The next example pertains to autoimmune diseases. This is a sample we obtained from a patient with type 1 diabetes. You can observe autoreactive pathogenic CD8 T cells identified using a tetramer against an insulin-related self-antigen. This is another example of tetramer application for a cancer antigen. Typically, the frequency of cancer antigen-specific T cells is very low. Therefore, prior to tetramer analysis, we needed to increase them. We performed this by stimulating patient blood cells with a peptide derived from cancer in vitro. After a 2-week incubation, we were able to detect the antigen-specific T cells using the tetramer. The applications for MHC tetramers in various areas of research are expanding. In particular, tetramers have various applications in the field of cancer immunity. The development of interventions, such as the dendritic cell vaccine, and treatments that use inhibitors of immune checkpoint molecules has led to exponential growth in this field. Accordingly, we are now expanding our tetramer product offering to allow clinicians and researchers to evaluate more immune cells and other conditions. With that in mind, let me explain our current product line. We can provide it through our made-to-order services. We have a fairly large product range, but there are 3 main ways to obtain our MHC tetramers. First, you can search for the tetramer of interest on our website or in one of our brochures. If you are unable to find the tetramers you are interested in, you can place an order through our made-to-order service for tetramer synthesis. Additionally, you can build your own tetramer using our new do-it-yourself custom tetramer preparation kit, Quick Switch. Now you can construct any MHC tetramer that you want in your own laboratory. Here is a list of the MHC alleles that we currently offer. MHC can be divided into 2 main groups, Class 1 and Class 2, according to its molecular structure and function. Each class consists of several hundred MHC epitopes and classifications. These are the MHC alleles dominant in the Japanese and Chinese populations. They also cover 80 to 90% of the population in North America. You can search our tetramer products on our website or in our brochures. In case you cannot find what you want, please contact our support desk for more detailed information. We recently launched a do-it-yourself tetramer kit named Quick Switch. This kit includes MHC tetramer preloaded with a placeholder peptide. You can exchange this preloaded peptide for your own peptide in the presence of the peptide exchange factors. It is very easy and ready to use. We currently offer this kit with MHC allele HLA, A2, A11, A24, and mouse allele H2KB, but we are expanding our MHC coverage in the Quick Switch range. Here is an example of a Quick Switch exchange tetramer. On the right is the data obtained using a classic tetramer. The middle is data obtained from a Quick Switch exchange tetramer. You can see that the results from the Quick Switch tetramer are comparable with those of our standard tetramer. This slide shows a global list of publications where researchers used MHC tetramers. The number of publications using MHC tetramers is increasing worldwide, particularly in the USA, Europe, Japan, and China. Many studies have been conducted in Asia, focusing on patients with cancer, infection, and autoimmune diseases. Therefore, we strongly believe that there are many novel opportunities for the application of this MHC Tetramer technology in Asia. We have both a product catalog and short leaflet for our MHC Tetramer product range available. Please check our website or feel free to contact our support desk for your copy. This brings us to the end of our presentation, and in conclusion, we would like to show you the take-home message again. MHC tetramers are a useful tool for monitoring immune cell activity in various human and mouse diseases. The advantages of using MHC tetramers include reduced nonspecific binding, resulting in clearer data than those obtained through the products of our competitors. With more than 800 pre-designed items, made-to-order service, and DIY kit, we are sure to provide a solution that meets your needs. Finally, we would like to emphasize that tetramers provided by MBL are considered the industry gold standard. We are confident that we can provide extensive technical support to all researchers using or having an interest in MHC tetramers. This concludes our presentation, and I thank you for your time. If you have any questions or comments, please feel free to contact our support desk.